function and structure of ATP. What are the three parts of an ATP molecule? One person. I don't remember.
Did you say that?
You've got three phosphates, that's one part. Adenine.
And
Ribose. Yep, good. Structurally, where within that ATP molecule is the energy localized? In the phosphate groups, yeah, the N phosphate is a matter of fact. Okay, how does ATP work? How does it work? Here's my ATP molecule, my three phosphates. How do I get this energy to this molecule right here? There's the last phosphate. Breaks off and attaches. What's that called? Transferring this phosphate from ATP to some other molecule.
We did it last year.
Phosphorylation, yes. Okay, importance of enzyme for cell reactions. What do enzymes do?
They inhibit, catalyze.
Speed up reaction, reduces reaction.
Lower activation energy?
Lowers the activation energy, lowers the amount of energy necessary to get it going. They don't give energy to a reaction. They just lower the energy needs of the system. Properties of enzymes. What are some things that we said, generalities about all enzymes?
They don't die.
They can be reused. They're a catalyst. What else?
They're specific.
They're highly specific. That means they're only used for a particular type of substrate. What else?
There's three.
What kind of molecule are they?
Proteins.
So proteins. What environmental conditions do we have to always examine when we're looking at?
Heat, concentration.
Temperature, concentration, and
Acidity, pH.
pH. Okay, induced fit model of an enzyme. What do we mean by induced fit?
Like form around the substrate.
Yeah, there's a region on the enzyme that kind of molds itself to the substrate. What's that region called?
Active site.
It's called the active site. We already said the conditions that change enzyme activity. All enzymes have an optimum pH, best temperature, best pH that they work in. You get too far either way, it's not going to work. I think that's what we're going to do in the lab Wednesday. They have a certain temperature where they work best. If the temperature is too low, what happens to the enzyme? Does it denature? No, it just slows the reaction down because the molecules are moving much slower. At high temperatures, excessively high temperatures, yes, the enzyme can be denatured. What does it mean to be denatured? Its shape is changed permanently so that it won't work. You know, you want to see a denatured protein, fry an egg. Egg white is protein. When it's denatured, you cook it. Look what happens to it. You can't get it back to the original form. That's the same thing that's happening to an enzyme, the same thing that happens to that egg white when it gets cooked. Okay, you said it has optimum pH, that means it works best in a certain pH. Certain concentrations of the enzyme work better. The higher the enzyme concentration, the faster it goes, to a point. At some point, increasing the concentration of the substrate or the enzyme won't make any difference. Why? Why is it that if you have an enzyme and a substrate, if you increase the amount of substrate or the amount of enzyme, it'll increase the rate of the reaction to a point, and at some point it won't go any faster. Why? Yeah, Vmax, it's going as fast as it possibly can. There's a certain amount of time they have to be together. And if the substrate's released very quickly and something else comes in absolutely immediately, it can't go any faster than that. Okay, and what's allosteric regulation? Last bullet of that.
That's where if the enzyme is making too many products, it will turn off the light switch.
Yeah, it's an on-off switch. What does it respond to? What's the feedback mechanism that turns it off? That's an inhibitor. We're talking about allosteric regulation, we're talking about inhibitors now. This is that temporary on-off switch mechanism, the allosteric site. What? No.
Quantity.
Of what?
Is this something that attaches to the back?
The products.
Oh.
When you get so much of the product floating around in the system, the product itself will inhibit will regulate it, it'll stop it from working. Oh, I mentioned it, and I didn't have it up here, I should have it up there. Inhibitors. What are inhibitors? They can either block the active site or change its shape. Where do they come from?
So,
They all come from the environment in some way. So that's the difference between an allosteric regulator and an inhibitor. An inhibitor is a poison from outside the cell. An allosteric regulator is something that's a product of the system itself that shuts it off, but it's temporary. So inhibitors most often are permanent. Allosteric regulators are temporary. Allosteric regulators are a product of the reactions. Inhibitors are from outside the cell. Okay, chapter 9. The equation, it's right up here. We've talked about this several times, oxidation and reduction. What are they? What's oxidation, what's reduction? Oxidation is,
No, it's the opposite reducing, so it's taking something away, taking another.
what's oxidation?
Like electrons are taken away from it.
So that's a loss of electrons by something. Reduction is a gain of electrons. Intermediate energy carriers. Somebody just said that. I don't know what these are. She's gone. What are the intermediate energy carriers that we talked about?
The
NADH and
FAD.
FAD.
Oh, FAD.
FAD.
Oh, just for cellular
Yeah.
respiration, yeah.
Those two B vitamins that we talked about. Okay. So what's their function?
carry protons.
Oh, yeah. they-
Protons and electrons from one place to another, whatever that place is.
It's
In
always-
cell respiration, where are they carrying it from? Where to where? From what region to what region?
Inside and the outside? Or, no.
From the matrix to the inner membrane.
So those considered a proton motive force?
No. The proton motive force is the energy that comes from the hydrogen ion concentration. Describe a mitochondria.
You could hear it.
What are the different regions?
You know how you could hear it.
Interfolds, which is the
Right
crystal.
here. Inner membrane, crucide, ATP synthase, matrix, outer membrane. You better know. I've drawn it for you several times. You saw them for the first time in chapter 6, and I redrew them in this chapter for you. Okay, compare aerobic and anaerobic processes. Aerobic processes
Require oxygen.
require oxygen, anaerobic don't. No oxygen, right. Fermentation.
This is really good.
What's fermentation do?
Acidic fermentation.
There are two kinds, acidic and alcoholic, and,
And it's when the oxygen is not present.
right, okay, so fermentation is just a process that enables glycolysis to continue in the absence of oxygen. That's the best way to know it. Okay, and this, in effect, is a diagram that I've shown you half a dozen times. Put it up one more time. What are the three parts? Glycolysis, citric acid or Krebs cycle. What's this?
There is phosphorus.
Oxidative phosphorylation. What goes in? Glucose. What goes in?
2 ATP, NAD.
What comes out?
4 ATP.
What goes here? What goes here? Acetates. What comes in, out, out? What's the waste product here? This is where the 6 carbon dioxide, 4 from here and 2 from up here. Okay, and we're gonna end up getting a total of 6 from here and 2 from here. This and this goes into here. What else comes in here? Oxygen. What comes out here?
34.
Oh, and ATP.
And here we have 8 NAD plus going in, 2 FAD going in. So in this one, I do want you to know numbers. This was 10 steps. This was in the cytosol. This was anaerobic. This was in the matrix. This was 10 more steps. This was, we had ATP synthase playing a role here, and we saw that it was in the Christi. Okay. So that's the kind of stuff you need to know. Any questions about those? Chapter 10, the equation. CO2 plus 12H2O yields C6H12O6, 6O2, 6H2O. Describe a chloroplast. Be able to draw it, be able to label it. I want you to be able to relate the processes to the organelle in which they're taking place. Structure and function. Location, raw materials, and products of the late reactions in Calvin cycle. You do it as the black box again. Light reaction, Calvin cycle.